This is a lecture that throws together infectious diseases from a lot of other systems of the body. I could make a lecture, and have in the past made more than one lecture out of all of these systems, but here what I've done is removed a lot of the individual diseases and pathogens, and in other cases I've chopped them down to make them fit in this one lecture. So there's still a lot, and you know how to go through and find the information you need. You've seen the kind of questions you're going to be asked on exam 4, so focus on that and you'll be in good shape. So let's get into it. We start with the oral cavity, and what we think about with the oral cavity is not normally the infectious diseases. There aren't major ones we normally think about in microbiology that preferentially affect the oral cavity itself. There are lots that affect multiple different mucosal surfaces, but not the oral cavity. What the oral cavity has that's unique is teeth and a tongue and exposure to food in the outside world. So the teeth are fascinating because they have this constantly evolving bacterial community. So there are specialized bacteria that colonize, that can colonize the enamel itself and form what we call plaque. And then there are secondary colonizers that can't really do that, but they can start growing on the surface of the plaque. And over the period of a day, we would normally expect all of those to appear on the teeth. And then if those aren't removed for whatever reason, either not brushing or not successfully brushing, the tertiary colonizers can take over. And these you could think of as the more efficient, more competitive bacteria. And these are the ones that typically cause disease that we think about. So some of the tertiary colonizers are the ones most likely to produce acids from dietary sugars. So these are the ones that are going to dissolve the enamel and lead to dental caries or cavities, and ultimately tooth loss. So other tertiary colonizers grow near the gums and lead to inflammation of the gums, and that kind of repeated inflammation of the gums ultimately can lead to dysfunction of the immune response in the gums, and that really is what periodontitis is. The soft tissue starts to decay, and it's complicated. One of my thesis students studied this for a semester, talked to some experts in the field. It was cool, but it's obvious that we still don't have a perfectly good handle on how this happens. But we do know that it starts with chronic inflammation that comes from having those gram-negative tertiary colonizers near the gums. And that constant inflammation eventually screws something up and leads to periodontitis. And that's what leads to tooth loss and everything else. So that's all I'm going to tell you about the oral microbiota. Right, so off we go to the rest of the digestive system. So I know that the mouth is part of the digestive system, but most of these diseases are going to affect the intestines, in some cases the stomach. And these are the diseases that best fit the icons I've been giving you, the intoxications, the inflammatory diseases, or the invasive diseases. So in some cases, like when we look at skin diseases, we have things that never cross the epithelium. They don't even damage the epithelial cells, but they cause an immune response. So that would be like a lot of the dermatophytes or the cutaneous mycoses. In the gut is not as well protected. So lots of different pathogens can affect the epithelia directly, and there's no dead cells on the surface to protect it. So a lot of these are going to interact with the epithelia directly. And they're either going to change the way the epithelial cells behave or attack the epithelial cells. In a lot of cases, it's hard to say whether a pathogen is invasive or not because it will get into the epithelial cells. And it's basically its choice whether it wants to go into deeper tissues or not, because the epithelial cells aren't necessarily designed to keep a cell in their cytoplasm. And invasive diseases in some of the other lectures have been more serious, but that isn't always the case with the digestive system. Diseases like cholera have a simple effect on the body. They get the epithelial cells to pump ions out into the lumen of the gut, and water follows. And that's all they really have to do, and that's enough to kill a person, through dehydration. So we have to take all of these kind of seriously and distinguish them from each other. So we start with the intoxications, and that would be food poisoning. And so Staphylococcus aureus is one of the common causes of food poisoning, and some people think it's the most common. I don't think we have good numbers on this because it typically limits itself. A typical case would last for a day or two, and while the person would be very sick and they wouldn't be able to work during that time, complications are not common. So you know about Staphylococcus aureus and the things it can do in the other tissues. What it does in food poisoning is it grows in food and it releases a toxin, a proteinaceous toxin, or a protein called an exotoxin. And it's heat stable. So boiling food doesn't remove this toxin. So somebody who's preparing food and maybe has a staphylococcal skin infection can contaminate the food with those bacteria, and then when they boil the food, they think the food is safe, and if they put the food in the refrigerator, sometimes Staphylococcus can grow at the low temperatures. So basically, it's hard to avoid Staphylococcus. So in other words, someone with an abscess or boil on the skin, or even someone carrying it in their nose, which a lot of us do carry Staphylococcus aureus in our nose. So if you're sneezing on food or anything like that, there's a chance of causing food poisoning that way. So I use a lot of bold with this because it is totally preventable. Sneeze shields and wearing gloves and not preparing food with open wounds on the hands could successfully prevent this. The next kind of food poisoning is caused by Clostridium perfringens, and this is the famous bacterium that causes gas gangrene, and it has the fastest doubling time of any bacterium that I have seen measured. So when this grows in a person's arm, it can double probably every 10 minutes. In beef gravy, in warm beef gravy, or I should say hot enough to just about burn a person, it can grow and double in 6 to 7 minutes. So luckily, when we eat food contaminated with Clostridium perfringens, it doesn't cause gangrene, it causes food poisoning. Again, 1 to 2 days of gastrointestinal symptoms. And beef gravy is the perfect environment for it. So that's the main thing to remember about Clostridium perfringens. Something I just want everyone to geek out on a little bit is that no bacterium can replicate its DNA in 6 to 8 minutes. It takes 40 minutes to copy its genome. So how can it possibly divide in 6 to 8 minutes if it takes 40 minutes to copy its genome? The answer is bacteria have a clever way of starting to copy their genome, and then they start copying the copies and then they start copying the copies of the copies. And so when the cell divides, each daughter cell inherits DNA that is partially copied already. It's got replication forks, many branching replication forks. And it's very clever. And if that didn't make sense to you, don't worry about it. It's just a thing biology nerds should know. Bacillus cereus causes a more complicated food poisoning just because there are two different toxins it makes and puts into food, and two different diseases that happen because of that. So there's the diarrheal form and the emetic form. Diarrheal form causes diarrhea. Emetic form mostly causes nausea and vomiting. And, well, the difference between them mostly is what food they grow on. And so starchy foods are typically going to lead to production of the heat-stable enterotoxin that causes the emetic form. And proteinaceous foods are typically going to cause formation of the heat-labile toxin. So cooking these foods would break down the toxin. Cooking this food would not break down the toxin because it's heat stable. And just like, well, these bacteria form endospores, and so it's hard to keep them out of food. It's hard to guarantee that your food is not contaminated with endospores. In fact, you can't. You have to assume your food has some endospores in it when you start cooking. And you pretty much have to assume some endospores survive the cooking process. So if endospores are going to ruin your day, I don't know what to tell you. What we can do is minimize this. And realistically, this is what food handling temperatures are supposed to do. So if you cook food, you cook it at a temperature that doesn't permit these pathogens to grow. It's as simple as that. Some of them die, others just can't grow. If you refrigerate food, none of these are going to grow quickly. It's when food is kept at like body temperature for a long time, and then put in the fridge and then brought back out to body temperature, that's when you can get these bacteria growing and reaching populations where they're making enough toxin to make people sick. That's the basic thing, is they need to be at some kind of critical mass for that toxin to affect us. And so either contaminating the food from a wound for Staphylococcus aureus, or just accidentally growing these bacteria by mismanaging the temperatures. That's what happens with Bacillus cereus and Clostridium perfringens. Also, I should point out, these food poisonings are all caused by the most durable bacteria we have. So the two different genera of endospore formers are Clostridium and Bacillus. There are a few others, but they don't cause diseases we care about. And then the most durable non-endospore former is Staphylococcus. So you could think of food poisoning as being caused by durable bacteria. And your job in the kitchen is to prevent them from having the temperatures they want to grow at. Okay, so with that, I'll leave this to you. This is a good thing to remember. Food poisoning is not an infection. You don't need to treat food poisoning with antibiotics. And doctors typically know this, just remind them. So we will keep going with intestinal pathogens in the next video.